Question 1. What did we discuss, ma? We have begun with cell discoveries. 3 basic discoveries of Robert Hooke has discovered. What did he discover? Dead cells. Robert Brown? Nucleus. In what cells? Root cell of orchids. Okay. And one more discovery, Anton van Leeuwenhoek, he has discovered living cells, which he named it as animalcules, with the development of lens, focused lens, and he is the father of microscopy. After which we have dealt with cell theory, proposed by 2 major scientists. Who are they? Sladen and Schwann. Of them, who is botanist and who is zoologist? Botanist, Schwann is zoologist. Fine, right. Sladen, in which year he has proposed? 1838. What did he propose? All plants are composed of cells and products of cells. Schwann, what did he prove? That all plant bodies and animal bodies are also composed of cells, but then the limiting boundary of an animal cell is cell membrane. Of plant cell is cell wall. Clear, right? And then both of them put together, they failed in giving one point. What is that? How new cells are formed. That is being said by Rudolf Virchow in the year 1855 under the concept omnis cellula e cellula. Clear, right? So with this we are done with cell theory, which is basic and known to everyone. But then what is important? What are the exceptions of cell theory? What are the exceptions of cell theory, ma? Viruses, viroids, prions, lichens, rhizopus, which are multinucleated conditions, sieve tube cells, which are enucleated conditions. So to tracheids, very good. To know the exceptions of cell theory, first you have to be clear on the note that what are the components of cell. What are the major components of one single cell? Cell membrane, cytoplasm and nucleus. Nucleus, cell membrane and cytoplasm. These are the components of a cell. Dani manam cell antam. Okay. Anything which is deviating from that is not considered as cell. To that, cell theory is not applicable. Clear, right? With this, cell theory is clear to us. And then we have moved to prokaryotic cell. Yes or no, ma? Prokaryotic cell, we have started from the outermost complex, which is addressed as cell envelope. How many layered it is?
3 layers.
3 layered. Outermost layer is known as glycocalyx. Outermost layer is glycocalyx. Glycocalyx, if polysaccharides are very tightly arranged, what do you call it as?
Capsule.
Capsule. If it is loosely arranged?
Slime layer.
If the outermost layer is capsule, for what is it useful? To hide from the host immune system or to protect itself from host immune system. If it is loosely arranged like slime layer, yeah, it prevents from the loss of water and mineral nutrients. Clear, right, ma, till here? Glycocalyx is clear to us. So bacteria, based on where it is staying, it will decide whether to have capsule or whether to have slime layer. After that, next layer is what? Cell wall. Cell wall is majorly to give a shape and to protect the cell. Yes or no? Cell wall composition is majorly? Cell wall composition. Cell wall composition of bacteria. Very good. Peptidoglycan. Peptidoglycan is the major composition of cell wall of bacteria. But then, based on the compositions or concentration of peptidoglycan and other components like what? Teichoic acids, murin. Okay, based on this we have decided whether it is gram positive or gram negative. Now one question to you. You have observed the differences between gram positive and gram negative. Amongst these two, tell me which bacteria is more pathogenic. Tell me why. I have told you both the things. Okay, I will give you one example. That when we have learned the gram staining process, in the gram staining process we have observed that the cell wall layers, see here, peptidoglycan is rich in gram positive bacteria, which is around 80%, and it is very 10 to 20% of peptidoglycan is present in gram negative bacteria. So here, when the gram staining experiment is done, gram positive absorbs more stain of crystal violet or gram negative? Why? Because it has high concentration of peptidoglycan. Gram-negative bacteria will not absorb because it has high amount of polysaccharides and lipids, which are unsaturated lipids. So, it will not absorb the dye too much, okay. It will destain when it is treated with alcohol and iodine mordant, and it will stain up again when safranin is added, okay. So, now tell me amongst these two bacteria which can easily survive in any environment? Gram-negative, right, okay. It is staining with violet, crystal violet, and the stain is being removed, and again it is restaining, whereas gram-positive, once it is absorbed, it is not losing the stain. So, it is more prone to cell wall antibiotics like penicillin. These are not that prone, fine, right, gram-negative bacteria. Now, you tell me, gram-positive bacteria is more pathogenic or gram-negative bacteria? Gram-negative bacteria is more pathogenic. Now, we understood the reason why. For every statement which is written in the NCERT, you have to know the reason why. Then only you can answer statement based questions and assertion reason questions. Okay. Just reading on to the NCERT will not help you out in the statement based and assertion reasoning. You have to know the reason why. Fine, right, for every statement in the NCERT. So, after this what we have learnt about the composition of the cell wall. Yeah, cell wall is done, and then cell membrane. Cell membrane is the third layer of the cell envelope. Inclusions which are present on the cell membrane are known as what? Mesosomes. These are the inclusions of cell membrane. They are present either in the form of vesicles, tubules or lamellae. What are the major functions of mesosomes? Can anybody tell me? Respiration, secretion, increases surface area, cell wall formation, daughter cell formation. Okay, these are the major functions of mesosomes. Amongst all this, respiratory activity is the major function because it is releasing the respiratory enzymes. So, that is the reason mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Clear, right? Any doubts anywhere? Yeah. So, this is about mesosomes functions. And in few bacteria like blue-green algae and cyanobacteria, mesosomes are colored pigments. They consist of the colored pigments known as what? Chromatophores, which also consists of the photosystems, okay, which will aid in the photosynthetic activity. Fine, right? Examples of cyanobacteria include nostoc, anabina and oscillatoria. Yeah, and then we have also looked into the cytoplasmic constituents, genetic material and non-membrane bound organelles. Genetic material is what type ma? Double stranded circular. Fine, right. Apart from the genetic material, we have genomic DNA and non-genomic DNA. Non-genomic DNA also known as plasmid DNA. Genomic DNA will take care of metabolic activities of a cell. Okay. Non-genomic DNA or plasmid DNA will have some specific functions to the bacteria. First is specific phenotypic character it will exhibit. Second is sexual recombination. Third is antibiotic resistance. Okay. One type of sexual recombination it can facilitate is conjugation. When it comes to non-membrane bound organelles, first is ribosomes. These are the common non-membrane bound organelles both in prokaryotic cell and in eukaryotic cell. Ribosomes are the common non-membrane bound organelles that are present. But then the sedimentation coefficient is different. In prokaryotic cell, the sedimentation coefficient is 70S, larger subunit is 50S, smaller subunit is 30S. Whereas in eukaryotic cell, larger subunit is 60S, smaller subunit is 40S. And what type of ribosomes are present in eukaryotic cells?
80S.
Both 70S and 80S. Because even in eukaryotic cell, you have chloroplast and mitochondria. Inside chloroplast and mitochondria, what type of ribosomes are present?
70S.
Remember, in prokaryotic cells, you have 70S ribosomes. In eukaryotic cells, you have 70S and 80S ribosomes. 80S are present in the cytoplasm of eukaryotic cells. 70S are present in chloroplast and mitochondria of eukaryotic cells. Clear, right, ma? and you have to be clear on the subunits. What is the major larger subunit and what is the smaller subunit. And then comes inclusion bodies, which are storage bodies. They store the reserve food materials inside the prokaryotic cell, which includes phosphate granules, cyanophycean granules and sulfate granules. Clear, right? And then there is one peculiar phenomenon or the major difference between the ribosomes of prokaryotic cell and eukaryotic cell. That is, during the time of translation, what happens is, over a single mRNA, only one ribosomal subunit will come and bind. That is known as monosome. That gives only one protein expression, which is known as monocistronic expression in a eukaryotic cell. Whereas in a prokaryotic cell, over one single mRNA, you can see multiple ribosomes that are attached. Multiple ribosomes, okay, which is known as polyribosome condition, also known as polysome, and this gives out what expression, ma? Polycistronic expression. Clear, right? Polycistronic expression, monocistronic expression, polysome and monosome, very important features. Fine, right? And then about non-motile structures like pili and fimbriae. These are the non-motile structures. Pili, as I told you, it also helps in attachment to the substratum and conjugation between F plus strain and F minus strain. Fimbriae are the bristle-like structures which are sprouting out of the cell wall, not cell membrane, okay, and they aid only in attachment to the substratum. Yeah. So the recap of entire prokaryotic cell has went on with a 2x speed. I think this is a good revision for the students who has listened the first class. To new joiners, you can once again go through the class. Fine, right? I hope 80% of the concept, I think it is clear to you. So I will give you 1 minute of time. Just go on through the class notes and then we will be starting about eukaryotic cell. Once if you say you are done, I will start. Done? Shall we start? One more thing to highlight for you when at the time of revision. Okay, whenever you are going through cell theory and cell discoveries, highlight on the scientists and what are the points they have given out. On the advanced cell theory, that is by Rudolf Virchow, Omnicellula, Ecellulae, and when you are going through the prokaryotic cell, please highlight on the types of ribosome, on polysomic condition and on glycocalyx composition and on mesosomes specially and on motile non-motile structures. What is the only motile structure that we have learnt in prokaryotic cell? Flagella. What is the protein that is present there? Flagellin. What is the microtubular arrangement? These are the points you have to highlight whenever you are going through revision. Fine, right? Now look at the eukaryotic cell. We have two types of eukaryotic cells. One is plant cell and animal cell. First, look into all the organelles, limiting boundaries, outer boundary of plant cell, and then about animal cell, and then we will go on the differences between the plant cell and animal cell. Look at here firstly, in a plant cell, the outermost boundary is what? Cell wall. Cell wall usually you will call it as dead, but no, it is living and completely permeable. Okay, and then in between two cell walls, the cells are being connected with a living component called as plasmodesmata. Somewhere here it will be. Yeah, here, plasmodesmatal connections. In between the cell wall you will have what connections? Plasmodesmatal connections. From one cell to other cell. How they are connected? By plasmodesmatal connections. These plasmodesmatal connections has one protein pigment inside it. It is a protein filament, you can say proteinaceous filament, which will extend the connections. This is known as desmotubule. Desmotubule, which is the living component in the cell wall, we will call it as. Okay. Cell wall, how cell walls of two different cells are connected? Through plasmodesmata. Inside plasmodesmata, what is the proteinaceous filament you have? Desmotubules. Yes, ma? Chappu. Okay. And then next to the cell wall you will have cell membrane. Okay. Outermost boundary is cell wall. Next to the cell wall having cell membrane. Now when cytoplasm is considered, cytoplasm is the main arena of all cellular activities. Okay, that is common in a prokaryotic cell, in a eukaryotic cell. Cytoplasm is the main arena of all the cellular activities. But then you can see active compartmentalization. In prokaryotic cell there is no active compartmentalization of cytoplasm. Here you can see the active compartmentalization, where you can isolate the organelles based on the number of membranes it have. Okay, the only non-membrane bound organelle you can see in a plant cell is ribosomes. Ribosomes are the only non-membrane bound organelles. This you can directly note it down in your NCERT also. Same diagram you have, right? And then single membrane bound organelles. Always remember, ma, single membrane bound organelles, whenever it comes into your mind, remember about endomembrane system. First, endoplasmic reticulum. Two types of endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum, followed by Golgi bodies, followed by lysosomes, followed by vacuole. Fine, right? Endoplasmic reticulum of two types, smooth and rough, followed by Golgi bodies, lysosomes, vacuoles. These four organelles are placed under endomembrane system because the functions of all these organelles are coordinated. Be here all of you, okay? The functions of all these organelles are coordinated. Now you place it here, single membrane bound organelles. First, smooth endoplasmic reticulum, rough endoplasmic reticulum. Okay, what is endoplasmic reticulum over here? Surrounding the nuclear envelope, you can see some extended network like structure here. This extended network or reticulum of tubular structures we will call. What are these structures? Tubular structures which are in the form of network. So network or reticulum of tubular structures are called as endoplasmic reticulums. Above this ER, if ribosomes are present, you call it as what? Rough endoplasmic reticulum, whose major role is what? Synthesizing proteins. Smooth endoplasmic reticulum major role is what? Synthesizing lipids. And it also helps in detoxification of drugs, and it also helps in calcium ion concentration, okay, to modulate the calcium ion concentration inside a cell. This is about RER and SER, followed by Golgi bodies, which are again single membrane bound organelles. These two are single membrane bound organelles. Followed by Golgi bodies, what do we have, ma? Lysosomes. Yeah, here. And then vacuoles. See here in a plant cell, vacuole itself occupies 90% of the plant cell volume, okay, 90% of plant cell volume, and vacuole is surrounded by membrane which is known as tonoplast. Fine, right? Okay, it is surrounded by what? Tonoplast, and it occupies 90% of plant cells volume. This is not the case with animal cell. Along with this, we also do have bound organelles called as microbodies. Microbodies includes peroxisomes, glyoxysomes, and spirosomes. Got an idea on non-membrane bound organelle? Only non-membrane bound organelle in plant cell is what, ma? Ribosomes. Single membrane bound organelles? Louder? Had lunch, no? Then, yeah. Endoplasmic reticulum? One is given here, peroxisomes, glyoxysomes and spirosomes also. Okay. And then double membrane bound organelles. Double membrane bound organelles include nucleus, mitochondria, chloroplast. Nucleus, mitochondria and chloroplast are double membrane bound organelles. You are clear with all the organelles, right? From a plant cell, from the diagram also. You are clear with labelings. You are clear with the primary activities that are being conducted by those organelles. Yes, no, ma? Any doubts anywhere? Yeah. Now, here we have something called as microtubule. Only microtubule is given in your diagram, but we have two more proteinaceous network structures called as microfilaments and intermediary filaments. Okay, microtubules, microfilaments, intermediary filaments. These three comes under cytoskeleton. What is cytoskeleton? Cyto means cell, skeleton is structure. These filamentous, proteinaceous filamentous structures, they will help in the intracellular transport, intercellular transport and to maintain the cell shape. Okay. What are those three structures? Microtubules. They come under cytoskeleton structures. A quick recap on a plant cell. Everybody drop your pens. Listen carefully. A quick recap. Plant cell outermost boundary is what, ma? Cell wall. Cell wall is connected. Two cell walls of two plant cells are connected via plasmodesmatal connections. These plasmodesmatal connections at the center will have desmotubule. And then next to the cell wall it has cell membrane or plasma membrane. Inside cytoplasm, all the cellular activities will take place, wherein only non-membrane bound organelle is ribosomes. What type of ribosomes are present? Two types. 80S is present in cytoplasm, 70S is present in chloroplast and mitochondria. Yeah, those ribosomes are also 80S. Very good. Okay. Over endoplasmic reticulum, we have ribosomes, right? How they are present, I will tell you when I am explaining endomembrane system. These ribosomes are also 80S. Those ribosomes which are present in cytoplasm are also 80S. Only those which are present in chloroplast and mitochondria are of 70S. Now you are clear, ma? Yeah. So here ribosomes you are clear with. Single membrane bound organelles? Occupying how much percentage of? Surrounded by what? Okay. Double membrane bound organelles? Chloroplast is? We have plastids actually. Plastids and three types of plastids. Can anybody tell me? Chloroplast, leucoplast, very good. Chromoplast. Okay. Leucoplast, chromoplast and chloroplast. And then cytoskeleton structures like intermediary filaments and microtubules. And then microbodies are also single membrane bond. There are smaller particles inside a cell. Okay, they include what? Peroxisomes, glyoxisomes, spherosomes. Clear? Shall we move on to the next slide? Anything to be noted down, you can note it please. If you are noting it in your NCERT also, it is fine. In your class notes also, it is fine. But a clear fragmentation on plant cells, non-membrane bound, single membrane bound, double membrane bound should be clear to you. Then only you can easily learn about each organelle. Okay?
And microbodies are
Cytoplasm law. I will tell you about that function at last. Okay? As of now, you have a primary idea on all the organelles. Done? Now, about animal cell. Okay. Animal cell, one example is taken, that is microvilli, which is present in your small intestine, one cell of microvilli. So, here if you see the microvilli projections, you can see in the small intestine, and the outermost limiting boundary is what? Plasma membrane or cell membrane is the outermost limiting boundary. And inside the cell, you have double membrane bound organelles like nucleus and mitochondria. Do you find any plastered here? No. Only two double membrane bound organelles, those are nucleus and mitochondria. Non-membrane bound organelles are two here. Very good. Ribosomes and centrioles. These are non-membrane bound organelles. Fine, right? And single membrane bound organelles, again the same pattern. First is what? Endoplasmic reticulum, smooth endoplasmic and rough endoplasmic reticulum, Golgi bodies, very good, lysosomes and vacuoles. Vacuoles are very small, they are not present as abundant that you see in the plant cell, they are present in smaller vesicles. Vacuoles are in the form of smaller vesicles. In few of the animal cells, they are absent also. That is a major difference between plant cell and animal cell. That is majorly because of the locomotory activity of the cells. Plant cells cannot move, so they have to store all the minerals, nutrients and water in the form of cell sap. Cell sap is present inside the vacuole. Okay. And here single membrane bound organelle, peroxisome is also there, glyoxisomes, spirosomes are also present. Okay. This is the clear fragmentation on what? Double membrane, single membrane, non-membrane bound organelles. One extra non-membrane bound organelle here is what? Centriole. Okay. Now, if it is done, we will move on. And then, so now first we learn about the first outer boundary, that is cell wall, and then we will learn about the limiting boundary of the animal cell membrane. So when it comes to cell wall beta, once we will remember the two kingdom system of classification, which is given by anybody? Very good, Carolus, Linnaeus. Carolus, Linnaeus, who has classified entire living organisms into just two kingdoms. What are they? Plant kingdom and animal kingdom. Major criteria is cell wall. Major criteria of his classification is cell wall. So when cell wall is considered, bacteria is kept under which kingdom? Plant kingdom only, no? In two kingdom system of classification, Bacteria is kept under plant kingdom because bacteria has cell wall. What is the composition of bacterial cell wall? Fungi is placed under plant kingdom because it also has cell wall. What is the composition of cell wall? Chitin has what units? NAG units, N-acetyl glucosamine units. Fine, right? Chitin is also known as fungal cellulose. Along with this, next comes what? Plant cell. What is the major composition of a plant cell, ma? Cellulose, hemicellulose, cellulose, hemicellulose, lignin and pectin. This is a composition of what? Plant cell wall. Algal cell wall. Algae cell also comes under plants only, but it is a primitive plant cell. Consists of galactans, mannans, xylenes, cellulose as well. These are different cell wall compositions of different cells: bacteria, fungi, algal cell and plant cell. If you are clear with this, then we will move on to primary, secondary, tertiary cell wall structures of plant. Done, ma? Done, right? Yeah. So here, first formed cell wall. To know about this, you have to know about the cell division, how it happens inside a plant cell. Observe at the end of telophase. At the end of telophase, how plant cell will be? Here at the end you will have two nucleus structures. Okay, again which is uncoiled, nucleolus, and two cells are formed. Now at the center, cytokinesis should happen. Telophase is the last phase of karyokinesis. Followed by this, what happens, ma? Cytokinesis. Listening. Here if you observe, at the center, the components which are released from Golgi bodies. At the center, completely inside this cell, cytoplasm will be present, right? Inside cytoplasm, Golgi bodies will be present, right? They will be releasing out the structures which are rich in calcium pectate, and magnesium pectate. This structure initially it is known as phragmoplast. Okay. This phragmoplast at the center, it will extend its structures. So as it is extending from the center to periphery, this mode of cell wall formation is known as centripetal cell wall formation. Centripetal mode. Fine, right? Once it is extended towards the periphery, okay, this structure is known as what? Middle lamella, or cell plate. Now you understood how this is formed? Drop your pens once, drop your pens, listen carefully. So at the end of telophase, you can see two nucleus structures which are uncoiled again. Okay, they are completely coiled with nucleolus. here at the center you can see the structure which is developed because of the synthesis from Golgi bodies. What is it rich in? Calcium pectate, magnesium pectate. At the center this is formed called as phragmoplast. This will extend towards the periphery. You will call it as what? Middle lamellae or cell plate. Fine, right. Once it is extended towards the corner, middle lamellae is formed. Middle lamellae is the first formed plant cell wall layer. Now here you see, if I can consider these two as cells. So the first layer which is formed is what ma? Here at the center, middle lamellae is formed. Next to the middle lamellae, that is inner to the middle lamellae, primary cell wall is formed. Inner to the primary cell wall, secondary cell wall is formed. I will draw a diagram which makes you more clear. Till then you can copy this. Done ma? Listen here. So at the center can you see the middle lamella? In between two cells, this is a plant cell. Consider this as what? Cell membrane. Okay. In between two cells, the first formed layer is middle lamella, which is rich in calcium pectate and magnesium pectate. This thing is clear to you. Next to the middle lamella, primary cell wall is formed. Okay. Primary cell wall is formed. Primary cell wall is thin. It is extensible. Okay. It will expand. As the cell is growing, the primary cell wall also expands. Its major composition is parenchyma. It is parenchymatous. Fine, right? It is parenchymatous. It will expand its structures. Okay. Primary cell wall will extend. Next, inner to the primary cell wall, secondary cell wall will form. Primary cell wall is single layered, but secondary cell wall is 2 to 3 layered. As the secondary cell wall increases in its size, as the cell increases in its size, what happens is the primary cell wall structure will disintegrate. Okay. Primary cell wall structure disintegrates as the cell increase in its size. Understood? Secondary cell wall is 2 to 3 layered, and it will be thick and tough because of suberin and lignin depositions. Everybody, I hope you are clear with this, right? Middle lamella, primary cell wall, secondary cell wall. In few tough plants like tall plants and all, tertiary cell wall is also formed inner to the secondary cell wall. Now, if you have to observe the cell wall layers, close to the cell membrane, which layer is present? Close to the cell membrane, secondary cell wall layer is present. Next to the secondary cell wall layer, primary cell wall is present. Next to that, what is present? Cell membrane is present. take this, very important. You can drink water, no problem.
Numerator. So numerator plus denominator is what? A plus B. Correct, no? A plus B. Numerator is what? A minus B. And row, dividend row. Okay. So numerator
Done?
also. Numerator.
This right? Shall we move on to plasma membrane model? Okay. So next is cell membrane. The structure of cell membrane became evident with the discovery of electron microscopy from 1950s. As such, many models have been discovered, like sandwich model of cell membrane, unit membrane model of cell membrane. Okay, like that we have got many membrane models, but the widely accepted model is fluid mosaic model, which is being proposed by Singer and Nicholson in the year 1972. Okay, in the year 1972, Singer and Nicholson has proposed fluid mosaic model. In this fluid mosaic model, what is the cell that they have considered? A mature RBC or a mature erythrocyte. Why did they consider a mature RBC, ma? Can anybody answer? Because it is enucleated, they can clearly learn the extended structure of the cell membrane or plasma membrane. Okay. So, when RBC is taken, what is the major composition of cell membrane here? It has proteins, lipids, carbohydrates. How much percentage of lipids and proteins are present? It is 52% of proteins and 40% of lipids, just 5 to 8% of carbohydrates. Okay. First, proteins. We have three types of proteins that are present now. First is peripheral proteins. Second, transmembrane proteins. Third, integral proteins, which are also known as tunnel proteins. Okay. And then we have lipids. Lipids will have two ends here, beta. One is hydrophilic. Very good. Hydrophilic head or tail? Hydrophilic head. Very good. Carbohydrates are present in two forms as glycolipids and glycoproteins. So now we are clear with the composition of cell membrane. What are present, what are different types of them. Look into the lipids first. It has hydrophilic head. These are the small globules, can you see? Even in your NCERT diagrams, these are the hydrophilic heads which are towards the outer side of the cell membrane. That means outer environment of the cell membrane, what are present? hydrophilic heads. Okay. Towards inner side what are present? Hydrophobic tails. Hydrophobic. They have fear of water molecules. So this hydrophobic tails are prevented from the water aqueous environment outside with what? Hydrophilic heads. Hydrophilic heads are considered as polar heads. Hydrophilic head is polar. Hydrophobic tail is non-polar end. So the non-polar hydrophobic tails are protected from the aqueous environment outside by hydrophilic polar heads. Understood everyone? Should I repeat it once again? Hydrophobic non-polar tails are protected from the aqueous environment outside by what? Hydrophilic polar heads. Now we are clear with ma? Yeah. So this is about what? Lipid composition. The major types of lipids that are present is phospholipids. Best example is lecithin. You will learn in biomolecules as well. Okay, the major composition of phospholipids is, phospholipids. One example of phospholipids is lecithin. We have many types of lipids actually, glycolipids, phospholipids, sphingolipids. Major type is phospholipids. Clear with lipids ma? Shall we move on to proteins? In proteins, if you see, we have three types of proteins. One is peripheral, transmembrane, integral proteins. Peripheral proteins are present on the either sides of the plasma membrane over the surface. Integral proteins or tunnel proteins, they will transpan both the sides of plasma membrane. Can you see here? Integral protein or tunnel protein, it is transpanning both the sides of plasma membrane. Whereas transmembrane proteins, these transmembrane proteins will transpan any of the side of plasma membrane. Can you see here? This is transmembrane protein, transmembrane protein. Okay? Transmembrane protein, it must transpan any of the sides, or if it is transpanning both the sides also, okay. But the prerequisite is it has to transpan one side of the plasma membrane. Okay. Now can I tell all integral proteins are transmembrane proteins? Can I say that?
No.
Why yes, why no? Integral proteins are transpanning both the sides, no? So I can say all integral proteins are transmembrane proteins. But not all transmembrane proteins are integral proteins. Major statement. If you are clear with lipids and proteins of plasma membrane, we will be moving to carbohydrate part. Done? Yeah. Now carbohydrates, if you see, the major role of carbohydrates is in the plasma membrane, they will help in cell to cell interaction in animal cells for building up of tissues. All cells should be together, right? So it will build up cell to cell interactions. Here carbohydrates are present in two forms, glycolipids and glycoproteins. Glucose along with lipid units is known as glycolipids. Glucose along with protein units is glycoproteins. Again they are being secreted from Golgi bodies itself. How? I will tell you in endomembrane system right now. But as of now, remember this point. So this is a complete idea on lipids, proteins and carbohydrates. If you are done with this, I will give you two major statements which are being asked many times in statement based question. First is how the lipid is present. You know, all the cells will be moving, they are motile, okay, based on their functions. They are not static or stable, right, inside a body of living organism. So the cell membrane, as its major composition is lipids, it also has cholesterol, okay, to maintain the fluid nature of plasma membrane. The movement of plasma membrane will be like this. That movement is known as quasi-fluid nature of lipids. Okay. This quasi-fluid nature of lipids will allow the lateral movement of proteins. Okay, it will allow the lateral movement of proteins. This is major point. Okay, quasi-fluid nature of lipids will allow the lateral movement of proteins. That is how the metabolism runs. Second point, that how the proteins are present over the cell membrane here, ma? Can you see? This is like an ocean, okay, and proteins are present like an icebergs. So how proteins are present? As icebergs over the sea. Quasi-fluid nature of lipids or proteins? Lipids. Enables movement of proteins. Okay. What are present as icebergs on the sea? Proteins. These are the primary points that you have to know about the composition and about the lateral movement, on how proteins are present. Then we will move on to the functions of plasma membrane. If you are done with this slide, we will move to the functions. And then functions of plasma membrane. The primary function is cell growth. Next, formation of intercellular junctions, that is majorly because of carbohydrates that are present in between. Secretion, endocytosis, cell division. So pause this endocytosis. We will learn about transport of plasma membrane. We have two modes of transport majorly. One is passive transport.
Active transport.
Very good. And one more type is also there. Can anybody tell me, ma? Anybody? Third type of transport. Bulk transport. See, there will be different types of molecules that are being passing out in between cells. Okay, just imagine there is a big traffic jam in between cells that happens. Different molecules will come, like how in the traffic junctions cycles will be there, autos will be there, cars will be there, okay, scooters will be there, bikes will be there. Same the case, we will have gaseous molecules, we will have non-polar molecules, we will have water molecules, we will have polar molecules, and we will have bulk molecules to be transported in between the cells. So based on the transport of different molecules and based on the utilization of energy at the time of transport or not, we have three types of transports now. First is passive, active, bulk transport. If you look into passive transport, very good. Passive itself says that it doesn't use any energy. The simplest form of energy is ATP, so we will say in the absence of ATP. That doesn't mean that is the only source of energy. That is the simplest form of energy. Rather than saying the absence of ATP, it will be more correct. It will not use any energy. There is no energy requirement. Okay, and this passive transport is of three types again, ma. What is that? Passive transport is of three types again. It is diffusion, facilitated diffusion, and osmosis. See, remember, all these are important. Okay, if you can look into the mock tests of NTA, from transport and plants also few questions have kept. Okay, this is a connected point with that. So listen carefully. First, diffusion, you know. What is diffusion? The gaseous molecules will diffuse from this corner to that. There is no special transport vehicle required. It will just diffuse. Okay. So, all the neutral solutes and gaseous molecules will be diffused via passive transport. They will be diffused in between two different cells. Clear right now? And then facilitated diffusion. The name itself is saying facilitated means what? Taking some help. Taking the help of either carrier proteins or channel proteins. Okay, it is taking the help of carrier proteins or channel proteins to transport the molecules. Okay, to transport the molecules. That is what, Nana? Facilitated diffusion. So, here if you see what molecules will be transported via facilitated diffusion is polar molecules. Polar molecules like amino acids and glucose. Clear till here? Any doubts anywhere? And then is osmosis. So, whenever we are discussing about drop sequence, I'll give you ample time to note down. Listen carefully. First understand the complete concept. Whenever you are going through the concepts of osmosis, reverse osmosis, you all know it is from low concentration to high concentration. But there will be a big doubt on what they are talking about concentration. Is it about the solute particles or is it about solvent particles? What is the low concentration they are talking about? Right. It depends on the question that he is giving. Okay. I will tell you, see, mostly it will be solvent only from low concentration to high concentration. For example, osmosis process only, plasmolysis. The concept is applicable for the pickle preparation also. If any cell, if it is placed into a hypertonic solution, okay, hypertonic solution, what happens, ma?
Shrinkage of protoplast.
Why? Anybody? Any language, no problem. Tell me the concept involved. Hypertonic solution. Here more amount of, let's say, salts only you say. Okay, high amount of salt or sugar concentration is present. When compared to here, salt or sugar concentrations, here it is less or more? Less, right? Here it is more. Okay, so always the transport is from less concentration to higher concentration, low to high concentration. So all the particles from the cell will be drained from the cell to the aqueous environment. That is the reason the cell will shrink. Second point, hypotonic solution. A cell is placed. Now you tell me what happens? Endosmosis occurs. Means what happens? The water molecules, the solvent molecules, from here they will enter into the cell. As the reason the cell will swell. Okay. So it depends on the concept where you are applying, whether it is solute or solvent. It is not always the solute, it is not always the solvent. But movement from low concentration to high concentration. For example, we are shifting from one classroom to the other. From the classroom 2, let us suppose 10 students have to come here. 10 students have to come here. Low concentration to high concentration. They will come fast, easily through the door. There is no requirement of much energy, pushing and pulling. But here in a class strength of 50, when you have to go to the classroom 2, there will be requirement of energy. Yes or no? You will push, pull, there will be some friction to go. Same the case. Okay. So if it is from low concentration to high concentration, it is said as passive transport, where there is no requirement of any energy. It is common. So that low concentration to high concentration is observed as concentration gradient. low concentration to high concentration is known as what? Across the concentration gradient. Because in your questions sometimes he will mention that the molecules are being transported across the concentration gradient. Across means low to high. Sometimes he will mention like against the concentration gradient, means high to low. So through osmosis, if I have to say, there will be some channel proteins that are present, majorly water molecules. One example if I have to give, water molecules which are being transported via aquaporins. So complete details of passive transport is given. If you are having any doubts, you can ask me, and then we will move to active. Out. Off చేయాలా? Done ma. Now you look into active transport. Active transport means against the concentration gradient. Means what ma? Anything, be it a solute, be it a solvent, if it is being transported from high concentration to low concentration, it requires energy. Okay? So requirement of energy means utilizes ATP. Then it is known as active transport. Bulk transport is of two types. Any molecules, if they are being transported in bulk, in huge, it is of two types, ma, endosmosis and exosmosis. Endosmosis and exosmosis. Endosmosis, if you observe, two types again, known as pinocytosis, phagocytosis. Okay, pinocytosis means cell drinking. Listen first. If into the cell, if small water droplets, liquid droplets are being absorbed, are being transported from low to high concentration in the form of vesicles, small vesicles. This is known as pinocytosis, and as these molecules are lipid droplets, this is known as cell drinking. Next is phagocytosis. If food particles in huge, if they are being transported into the cell, they are known as what? Phagocytosis, which is also addressed as cell eating, cell drinking and cell eating. Exosmosis, which is also referred as effagi. See, a cell will cleanse itself, okay. The digestive particles, dead particles inside a cell, they are being sent out of the cell, okay, via what? Effagi, exosmosis, which is also addressed as cell vomiting. These are different modes of transport of cell. First is what? Passive, active, bulk transport. Passive transport, three types: diffusion, facilitated diffusion, osmosis, Active transport against the concentration gradient, which requires the energy, that is ATP. Bulk transport of two types, endosmosis, exosmosis. Endosmosis two types, pinocytosis, phagocytosis. Pinocytosis for liquid droplets when they are entering inside the cell, bulk of liquid droplets, which is also known as cell drinking. Phagocytosis, cell eating. Exosmosis, cell vomiting. If we are done with the transport, then we will move on to endomembrane system. Functions of plasma membrane, clear ma? Cell growth, intercellular junctions because of carbohydrates that are present inside the cell, secretions, cell division and transport. Transport I have highlighted because few questions will be put in connection with transport in plants. Of course, transport in plants is deleted, but then all the concepts which are discussed here under transport is important ma. Important for your physiology also. One example of active transport you can say is sodium potassium pump. You will learn it in human physiology, no? Sodium potassium pump is one solid example of active transport. Done? Now, endomembrane system. As we have discussed, the first organelle of endomembrane system is what? Endoplasmic reticulum. Listen carefully, nothing to note down, I will tell you what to note, okay. Endoplasmic reticulum is what? Network or reticulum of tubular structures that are extending from nuclear envelope, okay. So, to this network of tubular structures, if ribosomes are present, it is known as rough endoplasmic reticulum. Listen, how the ribosomes are present? They are attached by a small protein called as ribofourin. Ribofourin is a small protein to which the larger subunit is 60S, right? As it is a eukaryotic cell and it is present in the cytoplasm of the eukaryotic cell. So, 60S larger subunit is attached by a ribofourin. Larger and smaller subunit is
40S.
Second is smooth endoplasmic reticulum. Remember, rough endoplasmic reticulum because of the presence of ribosomes. Ribosomes, wherever they are present, even in the prokaryotic cell, eukaryotic cell, inside chloroplast, inside mitochondria, wherever they are present, their only synthesis is proteins. Ribosomes will aid in the synthesis of proteins. So, RER will be synthesizing proteins which are naive, means inactive proteins. Smooth endoplasmic reticulum will be synthesizing lipids, not just lipids. If it is an animal cell, they will also synthesize steroid hormones. They will help in detoxification of drugs. They will maintain the calcium ion concentration, or you can say they will regulate the calcium ion concentration. You know how important calcium ion concentration is for muscle contraction movements. So, that is how they are controlling the muscle contraction and relaxation as well. By regulating what concentration? Calcium ion concentration. Clear, right? These are the new points you have to remember. Hold on. So, here you know they are synthesizing naive proteins and lipids. How they are being pumped to the Golgi bodies? They will be pumped to Golgi bodies in the form of vesicles. These vesicles are known as transitional vesicles. Not an issue. Listen here carefully, ma. So, here if you observe, this is nucleus, nuclear envelope. Extending to the nuclear envelope, you have the endoplasmic reticulum. like this. Okay, over which here ribosomal units are present. Okay, and these are being synthesizing the vesicles like this. Fine. These vesicles are being accepted by convex face of Golgi bodies. So, if you can see here clearly from the diagram, this is known as forming phase. This is facing endoplasmic reticulum, right? This is known as cis phase, forming phase. And you tell me, how does this look, concave or convex? Fine. How does this look? Concave. Mature phase, very good. Trans phase. Clear? What is that happening exactly inside the Golgi bodies? Here if you observe, when naive proteins and lipids are being synthesized, they are being accepted by cis phase of Golgi bodies. Inside the Golgi bodies, what happens is, it's okay ma'am, you can call from that door directly, from there directly. So here if you see, inside Golgi bodies, sugar molecules will be attached to these lipids and proteins. They will say packaging of proteins, packaging of lipids. What is packaging of proteins and lipids? Nothing but they will be adjoined to sugar molecules. One simple example I will tell you. You are making gulab jamuns at home. For that you need a sugar syrup. Okay. Whenever you touch the sugar syrup or jaggery syrup, how it will be? It will be little sticky and it comes closer, right? That's it is happening here. Proteins and lipids, they will combine together with the attachment of sugars. They can be glucose, they can be mannose, they can be fructose, any glucose units, any sugar particles. So that what are formed at the end? Glycoproteins, glycolipids, and some vesicles will be released out here. These vesicles will be rich in hydrolytic enzymes. What are hydrolytic enzymes? Can anybody tell me? So here they will be synthesizing what? Glycoproteins. Glyco, sugar units, proteins, glycoproteins. They will be sent to intracellular targets, extracellular targets also towards the plasma membrane. We have learnt, no? In plasma membrane 5 to 8% of carbohydrates will be there, that is in the form of glycoproteins and glycolipids. Okay, so these are being sent to plasma membrane. Next, some vesicles are formed. These vesicles are rich in hydrolytic enzymes. Tell me, what are hydrolytic enzymes? Lytic, remember all the time whenever you listen the term lysis in biology means breaking out. Okay, hydrolytic enzymes. The enzymes which will break out many biomolecules. Okay, it includes carbohydrases, proteases, lipases, hydrolases. Fine, right? And remember one more point, the pH of these vesicles is very low, means they are highly concentrated. The concentration is high. These vesicles which are being released from the mature phase of Golgi bodies are known as lysosomes. Okay, they are rich in, lysosomes are rich in hydrolytic enzymes. High proton concentration means pH is low. Lysosomes, based on their functions, we have different types of lysosomes. Okay, Different types of lysosomes include, first is primary lysosomes. Primary lysosomes include those vesicles which are just released from mature phase of Golgi bodies. If these primary lysosomes are attached to any digestive particle inside the cell, there will be many biomolecules that are present. Let's say you have ate nice dum biryani and you have got all the particles of proteins, fats, everything rich in your cells. Okay. Now the primary lysosome has attached to the digestive particles that are present inside the cell. Now that is considered as heterophagosome or secondary lysosome. Heterophagosome or secondary lysosome. Fine. If primary lysosome is attached to, after this digestion, the particles will be released, right? Some toxins will be released, right? Those are addressed as residual bodies. What are primary lysosomes? The vesicles which are just released from mature phase of Golgi bodies. If they are attached to any digestive particle like carbohydrates, proteins, lipids, what happens, ma? They will digest the particles, first thing, because they are rich in enzymes. They are addressed as heterophagosomes or secondary lysosomes, and the particles they are released are addressed as residual bodies. Fine, right? And here these primary lysosomes, if they are attached to any dead particles that are present inside the cell, you know, no organelle is permanent. The cell always cleanses the dead particles inside it or any toxins inside it. So when it is attached to any dead particles, they are known as autophagosomes. Okay, they are known as autophagosomes. So we have learnt about 4 types of lysosomes: primary, secondary, residual, autophagosomes. These are lysosomes. Now digestion is done inside a cell. Okay, dead organelles are also digested. All the toxins are present. Where should they dump this? Very good. They will be dumping this inside the vacuole, which is of 90% of cell's volume in a plant cell. So everything, all the toxins are being dumped to cell's vacuole. When they are dumped into this, this is addressed as cell sap. What is cell sap? Vacuole. Okay, and here inside the vacuole, the concentration is pH is less, that is concentrated, highly concentrated. High protons are present compared to the cytoplasm. So it is from, concentration is high here, it is low outside. Okay, but then solute particles are being transported here. Solvent is less here, solvent is more here. That means against the concentration gradient. Okay, solvent is less, solvent is more. So here how these toxins are being transported? Via active transport. Okay, it is via active transport. The molecules are being transported to the vacuole and it is dumped inside here, which is addressed as cell sap. Okay, everything is clear, right? Starting from endoplasmic reticulum, which are network of tubular structures synthesizing naive proteins, lipids. SCR is also involved in detoxification of drugs, synthesis of steroid hormones, regulation of calcium ion concentration. And here cis phase of Golgi, trans phase of Golgi. Cis is also known as forming, convex. Trans is also known as mature and concave. Inside Golgi bodies, what is happening? Packaging of lipids and proteins, which is nothing but, in simple terms, sugar molecules are being attached to lipids and proteins, which are being synthesized in the form of vesicle from mature phase. 2 points to be noted, ma. Those cells in which Golgi bodies are high amount of Golgi bodies which are present in high number in acrosome of a sperm cell and in the root cap cell of plants. High amount of Golgi bodies are present because in the acrosome high secretion is required to come and attach it to the ovum, and here in the root cap cell high secretions are required for positive geotropic movement. Okay. So, in these high amount of Golgi bodies are present. If you see in sieve tube cells, in mature RBC cells, Golgi bodies are absent, and in sperm cells of bryophytes and pteridophytes, because they are being transported via water. Water is the mode of transport of anthrozoids. Those are what? Sperm cells of bryophytes and pteridophytes. So, in what cells they are absent? Sperm cells of bryophytes and pteridophytes, mature RBC, sieve tube cells. Okay. This is the complete information about Golgi bodies. Glycoproteins, glycolipids are sent to plasma membrane, and here these vesicles which are rich in hydrolytic enzymes consist of what ma? Carbohydrases, proteases, lipases and hydrolases. They will be digesting the biomolecules. Those are just released from Golgi bodies known as attaching to digestive particle, attaching to any dead particles. After digestion the particles which are released, all the toxins, how they are dumped to vacuole via, because it is against the concentration. This is the complete story of endomembrane system. We are just left over with cytoskeleton, cilia, flagella, double membrane bound organelles. Next class we will do this. So, to boost your confidence, let us solve out few objective. Try out this one. Quick. Very good. But if you gisp out there, I can't listen. Be louder. Like how you can hear me clearly, no? Like that. Tell me your options.
One.
How many? One. Okay. How many? Two. Oh, everybody one. Unanimous. Let's see. RER. SCR. Golgi bodies. Lysosomes. That's it. Easy, no? This is how you need to apply. Yeah, this you need some idea. Wait a minute. This you can answer.
Just question will you?
Don't be in hurry. Think and answer. Fourth one? Sure? A. Osmosis without energy, which is option 1. Active transport? Energy is utilized. Passive transport? Done? Next. This you can answer. Second option, see this, lysosomes hydrolytic activity. Ribosomes,
protein synthesis,
smooth endoplasmic reticulum, centrioles,
formation of spindle,
formation of spindle. Tell me what is the right answer? That's it. Next. See, as this is basic, I have not included much of statement based questions, but you will get in further classes. You can answer that also. Think and answer. You can do it.
Square A minus sin square B.
Good. Good. It is about cell wall, right, ma? Provides shape to the cell? Protects cell from the mechanical damage? Helps cell to cell interaction? Provides barrier to undesirable macromolecules and imbibes water as well. This you can't. Tomorrow you will answer that. This.
Correct. So,
Wait, I want the answer from Augustine. Which one? Okay, we'll see. The shape of cells may vary with function they perform. Yes. Human RBC 7 micrometers in diameter. True. Cytoplasm is main arena of cellular activities. Various chemical reactions occur in cytoplasm to keep the cell in living state. If there are no chemical reactions that are happening in cytoplasm, how come the cell is in living state? It is dead, right? If there is no activity inside the cell, so all are correct. Yeah, that's all with today's questions, ma. Tomorrow we will solve out few more questions on this after the class, okay, and then we will also look into the DPP tomorrow. If you are having any doubt on cell wall, firstly on plant cell, on animal cell, on cell wall, on cell membrane, and then on endomembrane system. On the activities of 4 organelles we have learnt: endoplasmic reticulum, Golgi bodies, lysosomes, vacuoles. Any doubts in this concept till now, you can ask me. Okay? Go through it, open your NCERT's class notes, objective books.